look at classification and the naming of enzymes. Today we'll continue with the classification of enzymes, and then after that, we'll take a look at coenzymes and cofactors. All right, so as I was saying, we'll continue today with classification of enzymes. Today we're looking at classification of enzymes based on specificity to the substrate on which the enzyme is acting, and some other forms of specificity. So here we are seeing four main groups as far as specificity of an enzyme is concerned. And the first is absolute specificity. So what we say, absolute specific enzymes. These are enzymes that react with a single substrate. So only one substrate. Then we have group specific enzymes. These act on molecules that have some similarity, particularly in functional groups. And we have enzymes that are bond specific. So these enzymes will act on specific bonds in molecules. And then the fourth one we'll look at is stereospecificity, where an enzyme will be acting on a particular isomer of a molecule, either the D isomer or the L isomer. We'll see some examples of these. So let's dive in and take the first one, which is absolute specificity. Now, the first example we are looking at is an example which we saw earlier, and that is lactase. Lactase. We saw lactase as an enzyme involved in the breakdown of milk sugar, lactose. And now the name clearly should tell you that this enzyme is specific to lactose. That's why it's having the name lactase. If this enzyme was acting on any other substrate, then the name lactase will be misleading. So the name tells you that this enzyme is absolutely specific to the substrate lactose. And what you see on your screen, the reaction mechanism you see on your screen, is just to tell you that lactase is breaking down lactose. So you see at the bottom here, on the left bottom, we see lactase. And beside it, you see glutamate 1749. The only thing this is saying is that, you see, we saw earlier that enzymes are mostly proteins. So lactase is a protein. It's a big protein. big protein, composed of thousands of amino acids. So if we are looking at the amino acid sequence in the structure of lactase, that is, if we are counting the amino acids that are bonded to each other to give you that big molecule, we are counting from first amino acid, second, 3, 4, 5, 6, all the way into the thousands. At the 1749th position is glutamate. So this is telling you that this is actually the active center of lactase, because it is that glutamate which is having an attack on the glycosidic bond in the lactose molecule. So what you see here, D-lactose, you can see, lactose is a disaccharide consisting of glucose and galactose. So you see the lactase molecule, the enzyme, we are seeing on that glutamate, the side chain of the glutamate is what is showing here. And that side chain is what is attacking the glycosidic bond between the glucose and the galactose molecule that are bonded together. And that bond is broken. And when the bond is broken, you see first you have a glucose molecule that is released. And then you see the galactose molecule bonded to the enzyme. So you have that covalent bonding between the lactase molecule and the galactose molecule. That's an intermediate, galactosyl intermediate, which later the enzyme dissociates from that galactose molecule. So you have a free galactose molecule, and first you had the free glucose molecule. And so the enzyme will be free again to continue attacking other lactose molecules and be breaking them down. So that's the first example, lactase acting specifically on lactose, breaking it down into its components, glucose and galactose. The second we are looking at is glucokinase. Glucokinase. And this enzyme is also acting on only glucose. Only glucose. And what does it do? I think we mentioned in the first lecture that if you see the name of an enzyme with kinase in it, kinases are involved in the transfer of phosphate groups from one molecule to the other. So here you see this is a glucose molecule, and that is being phosphorylated to give you glucose 6-phosphate. And the enzyme that is acting in that process is glucokinase. And what is glucokinase doing? Glucokinase is transferring phosphate group from ATP onto the glucose molecule to give you glucose 6-phosphate. So once it gives out a phosphate group that binds to glucose to give you the glucose 6-phosphate, you'll be left with adenosine diphosphate, ADP. So that's another absolutely specific enzyme, glucokinase. The name also tells you it acts specifically on glucose, no other substrate. Now we move on to the second group the second group of enzymes based on specificity. This is group-specific enzymes. Now, group specificity, we saw earlier, means that an enzyme is acting on molecules with similarity in some functional group. So the enzyme we are seeing here is hexokinase. Hexokinase. And hexokinase is acting on hexosis. So the reaction we saw in the previous slide is the same reaction that we are seeing here, where you see glucose is being phosphorylated to give you glucose 6-phosphate. In the previous slide, we saw the enzyme which was acting in the process was glucokinase. So that enzyme was specific to glucose. Now we are seeing hexokinase. Now hexokinase means that it will be acting on all 6 carbon sugars. So not only glucose, but all 6 carbon sugars can be acting on fructose, can be acting on maltose. So all the 6 carbon sugars, if you go look at all those 6 carbon sugars, yes, hexokinase can be acting, but as a matter of fact, in the system, in the human system, it's actually an activity on glucose, except that you have hexokinase is present in all tissues, it's present in all cells, but you find glucokinase more in the liver and in the pancreatic cells. So glucokinase helps, much more in the regulation of glucose concentration in the blood. So we see on the right-hand side, what you see here is, the activity of hexokinase compared to the activity of glucokinase. And we see, hexokinase is more or less, at a constant concentration all along. But we see that glucokinase, actually shoots up, rises up. And the reason that is that anytime that you eat and you have the concentration of glucose rising in the blood, glucokinase concentration also rises in order to break down the glucose, bring the concentration to normal levels. But hexokinase is constant, it's always acting, and actually it will be acting much more in times of fasting. So for instance, when you are sleeping at night and you are eating, hexokinase will be acting in all cells, particularly in the brain cells as you are sleeping, so that there will still be, generation of energy to the tissues and the cells as you are sleeping. And at the same time, in a way to sort of, allow for the glucose levels to be at a certain constant rate. At that moment, you see glucokinase is much more in the liver. And so, and then I said also in the pancreatic cells, but particularly in the liver at that fasting moment, it will be involved in the breakdown of glucose molecules that are being released from glycogen, the glycogen storage in the liver, which is responsible to give energy to particularly the brain cells. So it's a way of allowing for enough energy for the brain cells. So see that the glucokinase levels, sort of they go low when you are in a fasting period, you are sleeping. And it's actually this knowledge which is applied in the technology of the fasting blood sugar testing, so that you see glucokinase levels, the activity of the enzyme is proportional to the increase in glucose concentration, or let's say the concentration of glucose in the blood, as you can see, and so that the activity of the enzyme will result into an analytical signal that can be recorded, to measure what we refer to as, the fasting blood sugar. All right. Now, before we move to the next, I would say you should quickly do some revision of amino acids. In those of you I taught in HND first year, we studied the structure of amino acids. If you've forgotten, you should remind yourself of these because it will help you to understand what we're talking about. I mean, the next thing we're going to talk about. And that is to say, particularly we focus on amino acids with side chains that are hydrophobic, and particularly the bulky ones. Because see, alanine is having a side chain of just a methyl group CH3. That's not so big. But after that, you see that side chain gets more and more complex with valine, isoleucine, leucine. See the side chains are getting longer and more complex. And then methionine. At the same time, you remember the three amino acids with aromatic side chains. These are phenylalanine, tyrosine, and tryptophan. Now, these amino acids also with these aromatic side chains are hydrophobic. These chains are hydrophobic, and we can see clearly that these side chains are bulky. They are as bulky as even these aliphatic side chains that are also hydrophobic. Now, having reminded yourself of these side chains, the next class of enzymes is still the group-specific enzymes, but the next example that we are taking after what we just looked at with the hexokinases, we're looking at pepsin. Now, pepsin acts on the peptide bonds that amino acids with bulky side chains are involved in. So, for instance, if you take phenylalanine, so this phenylalanine, the side chain is the ring structure. That's an aromatic side chain. And we see it forms peptide bond with 309 and forms a peptide bond also with lysine. Now, what pepsin does is, pepsin actually is involved in the breakdown of proteins that we refer to them as less digestible proteins because they contain these hydrophobic side chains. That means they are going to be more, or let's say they are going to be less soluble or more insoluble. So we see pepsin involved in the breakdown of such proteins, particularly breaking down the bonds that these amino acids with a bulky hydrophobic side chains are involved in, which means pepsin will come and break these bonds between phenylalanine and threonine and phenylalanine and lysine. So those are the bonds that pepsin will be involved in breaking down. And so it's group specific because we just saw that amino acids with the aromatic side chains were 3. So it means that even if you take only the amino acids with the aromatic side chains, you are already having 3 substrates. That is phenylalanine, tyrosine, tryptophan. Then if you take those other aliphatic side chains that are also hydrophobic that we just saw, like those of valine, leucine, isoleucine, those are more substrates also. So that is a group-specific enzyme, pepsin. Then we also see that trypsin will also be acting on peptide bonds that amino acids arginine and lysine form with their carboxylic acid group. The carboxyl, group side of the peptide bond of these amino acids. That is what trypsin will be involved in breaking down. And we see, the similarity between arginine and lysine is that they are having this extra amine group on the side chain. And so you see they are carrying a net charge, a net positive charge on the side chain. You see that. So that is also another group specificity. It's not only, it's not a single substrate. It's acting on arginine and lysine. And we see that there is, you have some similarity in the functional group, that in the side chain, you are having an extra amine group, which is giving that amino acid molecules a net positive charge. Now, before we move to the next, you should also quickly remind yourself of first year biochemistry of the alpha and beta configurations of monosaccharides. Remember when we're looking at the cyclic structures of monosaccharides, the Haworth perspective formulas. And we talk about the fact that the first carbon number 1 in the cyclic structures of the monosaccharides is what we refer to as the anomeric carbon. And on that anomeric carbon, you can have the groups rotating. So the hydrogen and the hydroxyl group can be rotating. And when you have the hydroxyl group pointing downwards, you have the alpha configuration. Then you have the beta configuration when you have the hydroxyl group pointing upwards. Great. Having done that revision, we move on to bond specificity, where we see the example alpha amylase, which hydrolyzes alpha glycosidic bonds, the alpha 1,4 glycosidic bonds. So we see up here at the top, we can see alpha 1,4 glycosidic bond between 2 glucose molecules, alpha 1,4 glycosidic bonds between 2 glucose molecules. Now that is the bond that alpha amylase can break down. So if you have glucose molecules, like these glucose molecules that are bonded together, you have maltose. Alpha amylase can break down this into two glucose molecules. And so those glucose molecules can then further go into, the breakdown to generate energy. But alpha amylase is not able to break down the beta 1,4 glycosidic bond. So you see, the bond is being formed with that hydroxyl group, which was, which we saw on carbon number 1. And you see here, the below, looking at the beta 1,4 glycosidic bond here, you see that the bond is being formed with a beta configuration of the monosaccharide glucose molecule. And alpha amylase is not able to break down this bond. A common example, I think, again, those of you who were in my biochemistry class in first year, I think I mentioned to you that you have these beta 1,4 glycosidic bonds in cellulose. What is, the main component of the woody part of plants. And so that is why, when you take in food, you have part of it which is being referred to as roughage, like, fiber, like a lot of fiber in foods and things like that. That we refer to as indigestible part of food. Yes, indigestible because our salivary amylase cannot break down the beta 1,4 glycosidic bonds in cellulose, which is the main component of fiber. But it can break down the alpha 1,4 glycosidic bonds, what you have in starch, what you have in glycogen, which is stored in the liver. You have the alpha 1,4 glycosidic bond, which alpha amylase can easily break down. And I made an example of, for instance, you see termites eating wood. The termite itself does not have the ability to break down the beta 1,4 glycosidic bonds that are in the cellulose of that wood that the termite is chewing. But it has a symbiotic relationship with a microorganism that is in the gut, in the hind gut of these termites, trichonympha, and trichonympha has the enzyme cellulase, and that cellulase can break down the beta 1,4 glycosidic bonds. So if you see termites chewing wood, really not the termite itself is getting any energy out of it, but it's that microorganism that is benefiting from that. It's a symbiotic relationship in that manner. So that is why if you can't just take wood and eat and you'll be satisfied. If you take wood, you eat, it will just come out because your amylase cannot break down the beta 1,4 glycosidic bonds in cellulose, which is the major component of the woody part of plants. Now we can move on to take a look at stereospecificity. Now here again, remind yourselves of the enantiomers, isomers of a particular molecule that are mirror images of each other. These are non-superimposable. So when we go ahead and we're looking at the metabolism of the biomolecules, when we get to metabolism of proteins, you realize that some oxidation process, the oxidation of amino acids, which is a process of the breakdown of amino acids, you find some enzymes, oxidases, that will act only on the L isomer of particular amino acids. So you can have L amino acid oxidase that will be acting on the L isomers, whilst you have D amino acid oxidase acting only on the D isomers. Now you even see, generally, the proteins that are in us, or let me just say proteins in biological systems are mostly in their L isomers. It's like that's the active form of the amino acids. So that even when you consume some proteins in food, particularly from plants, where you can get some D amino acids. When we're looking at the metabolism of proteins, you come to realize that those D amino acids are first converted to L amino acid before they continue the metabolic process. And these D and L isomers, they act like biological switches, like on and off. Like I was saying that the L amino acids are like the active forms of these amino acids. So when you have the L isomer, it signals on, it's active on. So a reaction can proceed. But if when you have the D isomer, then the signal is off. So a particular reaction will not proceed. And so this is one of the functions of these, L and D isomers. But we mostly find them as the L isomers in biological systems. You find a few D isomers, particularly in food when you consume from plants. But once it gets into the body and is undergoing metabolism, it gets converted to the L isomer before it continues in the metabolic process. Now we can take a practical example of this knowledge of enzymes that we are getting. So we take the enzyme acetylcholine esterase. This is an enzyme that we saw in our last lecture as a hydrolase. When we're looking at the classification of enzymes into the 6 major classes: oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. So when we go to hydrolases, I think the example we looked at was acetylcholine esterase. And acetylcholine esterase was breaking down acetylcholine into acetic acid and choline. Right. Now, acetylcholine is a neurotransmitter, sort of signals muscles to contract. So it's involved in that contraction and relaxation of muscles. And how does it act? When it attaches onto a muscle cell, the muscle, so acetylcholine attaches to muscle cells, that's to a muscle tissue, and the muscle contracts. And when it contracts, acetylcholine esterase then goes in to hydrolyze acetylcholine, meaning, to sort of dissociate it from the muscle cells. That way, once it hydrolyzes away, it means the muscle will relax, and then a next signal will come. Acetylcholine will come again, so that there will continue to be contraction, relaxation, contraction, relaxation. So the acetylcholine esterase comes there to just break down acetylcholine into acetic acid and choline. We saw that in a previous lecture. This molecule is having a choline part, which is on the right-hand side, and then the acetic part is on the left-hand side. And that is what you see is broken down. The acetic side is removed from the choline part for you to get the acetic acid and choline. All right. Now, knowing this process that acetylcholine is involved in, the contraction and relaxation of muscles, a drug developed from this knowledge is succinylcholine. Succinylcholine is a bigger molecule. If you pay attention to the structure of succinylcholine, you realize that it is, you can say it's a dimer of acetylcholine, like two acetylcholine molecules that have, bonded together, have a covalent bonding between them. If you pay attention to that, you see this. You have the choline ends at the left and right-hand side. You have the choline ends. And then you have at the center the acetic parts that are bonded between them. So you have that full molecule, succinylcholine, as a dimer of acetylcholine. Now, the enzyme acetylcholine esterase, just as it's able to hydrolyze acetylcholine into acetic acid and choline, it can also hydrolyze succinylcholine. It can break down succinylcholine, except that it can do that at a very slow pace. I mean, this is logical. If acetylcholine is breaking down, if acetylcholine esterase is breaking down acetylcholine, which is a smaller molecule, I mean, less energy will be needed to do that than if it is coming to break down succinylcholine, which is a bigger molecule. So succinylcholine is used as a muscle relaxant, which is used in minor surgeries, that when it is injected into the tissue where surgery is to be performed, because it attaches to the muscles and it will take a long time for acetylcholine esterase to break it down, for it to dissociate from the muscles, which means the muscle will still be, bonded, or say the acetylcholine, the succinylcholine will still be attached to the muscle cells. And so it will not have space for another acetylcholine molecule to come to give it a signal to contract again. So it means that the muscle is going to stay relaxed for a long time, because the muscle is not free for the next signal. The muscle will to dissociate away from the muscle cells. So you find the muscle is relaxed over the period of the surgery until the surgery is done. And then by that time, acetylcholine would have resumed its work, and acetylcholine esterase will continue to be dissociating the acetylcholine from the cells, and normal contraction relaxation process resumes. So that's a way of applying, such knowledge of enzymology, particularly with acetylcholine esterase being used as a muscle relaxant. Okay, so it was a very practical one. So that you know that you are not just studying these things like in a vacuum, but they are actually very practical things. Now we can continue to coenzymes and cofactors. Coenzymes and cofactors. We talked a bit about coenzymes and cofactors when we were looking at the terminologies in the first lecture, terminologies that will help you to understand our conversation in enzymology. Now we see here an apoenzyme, which is the protein part. We said enzymes are mostly proteins, so that's the protein part, but it's inactive. It gets active when the coenzyme, so either a coenzyme or cofactor, which is a non-protein part, sometimes being referred to as the prosthetic part of the enzyme. And it's an activator. That's an activator. So these, when it binds to that's a whole enzyme, the active form of the enzyme. And so now you see that active enzyme is having an interaction with a substrate, which is depicted red, can have such an interaction with the substrate. So the coenzyme, the cofactor, acts as an activator. It is required for the enzyme to be active. Now, we take cofactors. Remember, I mentioned that even though coenzymes, cofactors sometimes are used interchangeably, there is actually a distinction between them. Cofactors refer to inorganic metal ions that are involved in the functional structure of the proteins, the enzymes. So it's not only enzymes per se, but even if you take, many proteins, you see that you have these metal ions that are embedded in the structure, actually holding either a tertiary or quaternary structure of these proteins. That is the functional structure of these proteins. And it is the same thing to do with enzymes. You don't call them cofactors per se in other molecules, but in enzymes, that's how you refer to them, cofactors. And we see copper 2 ions. Here are cofactors to cytochrome oxidase. We see iron 2, iron 3 ions are also cofactors to cytochrome oxidase, catalase, and peroxidase. Potassium ion serves as cofactor to pyruvate kinase. I think we saw pyruvate kinase somewhere. So a kinase, you have seen that name again, will be involved in the transfer of phosphate group onto pyruvate. You see magnesium ions are serving as cofactors to hexokinase. We saw that a few minutes ago. Glucose 6 phosphatase. Well, I mean, the name will tell you, yes, it will be involved in a transfer of
phosphate group, maybe to come and give you glucose 1,6-biphosphates after you have here you have only one phosphate group. And then again, pyruvate kinase. Manganese ions are serving as cofactors to arginase ribonucleotide reductase. So if you are applying the naming and you see reductase, it tells you it's involved in reduction reaction. Molybdenum is a metal also involved as a cofactor with the enzyme dinitrogenase. Then you have nickel ions. These are cofactors to urease. We also saw urease involved in the hydrolysis of urea. Selenium is a cofactor to glutathione peroxidase. Zinc ion is a cofactor to carbonic anhydrase. Alcohol dehydrogenases. I think we saw dehydrogenases, and I mentioned that dehydrogenases are oxidoreductases. Carboxypeptidase. So as a peptide, that you have some transfer of carboxyl group out of that peptidase, or because it's carboxy, it will rather be bringing a carboxyl group there. You have carboxypeptidase A and B, so these are different forms of carboxypeptidases. But our focus is on these ions. It's on these ions. So you have these ions that are acting as cofactors to some enzymes. There is no need, sort of chewing this. It's just, for you to see that, yes, you have metal ions that act as some prosthetic part of enzymes, some proteins. They are required for the functional structure of the protein. So you just have to know that. I mean, you should notice that a lot of times you have some vitamins or even some food supplements with minerals. And if you go and look, what are those minerals? You see the minerals are these metal ions in them, because these metal ions are required for several functions in the body. Now we can move on to coenzymes. I mentioned that coenzymes are also prosthetic parts of the enzyme. It's a non-protein part, but these are organic molecules. The cofactors are inorganic metal ions, but these are organic molecules. And they are mostly some vitamin Bs. Mostly, you find them there. You see, as we go through now, you see that there are vitamins, some different forms of vitamin Bs that we are talking about. So we see biocytin, the first one, biocytin, the first coenzyme. And what does it do? It's involved in the transfer of, it's written there as CO2, but it's not carbon dioxide. That's a carboxyl group. The carboxyl group, when it is an ion, the carboxylic acid group is COOH, but without the hydrogen is COO. So transfer is, can be involved in transfer of a carboxyl group from one molecule to the other, or even removing the carboxyl group from that molecule. And what is the dietary precursor? So what it means, dietary precursor, is the vitamin that serves as a precursor to biocytin. In other words, the vitamin which is being consumed, the vitamin that you consume from food, is an inactive form of the coenzyme. So when you consume a vitamin, is inactive, that vitamin is converted to an active form. That's what becomes the coenzyme. So biotin. So biotin, I think is one of these vitamins. I think vitamin B7 or so, vitamin B7, and that is activated. Its active form is biotin. The next is coenzyme A. I think you've seen coenzyme A a lot of times in many biochemical reactions. When you look at least if you remember your glycolysis to Krebs cycle, you remember there is some coenzyme A along the line somewhere, and is involved in the transfer of acyl groups. So like acetic acid, like a residue of acetic acid, acyl groups. And the precursor to coenzyme A is actually, when you see the structure of the coenzyme A, you see that there are different components that are in it. We'll look at it and you see. But one major one is pantothenic acid. It's also one of the vitamin Bs. I think it's vitamin B5 also, vitamin B5. And then you have some other compounds, some nucleotide and some amino acid derivative. All of those are bonded together to give you coenzyme A. You have deoxyadenosyl-cobalamin. And we see there is written coenzyme B12 coming from vitamin B12, and is involved in the transfer of hydrides or hydrogen atoms and some alkyl groups. So it can be some methyl group like CH3 or ethyl, etc. You have flavin adenine dinucleotide. You've actually been seeing these in a lot, many reactions, like you see some reactions and then on the arrow is written FAD. That is flavin adenine dinucleotide, which is involved in the transfer of electrons. So that clearly tells you that FAD, flavin adenine dinucleotide, is involved in oxidation reduction reactions. And that is gotten from riboflavin. I mean, the name also, you see the flavin there. So it's coming from riboflavin. It's also one of these vitamin Bs, vitamin B2. You take some foods, you take some food supplements and things like that. You'll be seeing any of these vitamin Bs on them. Lipids also, involved in electron transfer, so oxidation reduction reactions, as well as the transfer of some acetic acid residues, like these acyl groups. And these can be synthesized in the body. That's why it's here as it's not required in diet. You have nicotinamide adenine dinucleotide. What you usually see as NAD. Sometimes you see NAD, NADH, NADPH, these different forms. Also involved in oxidation reduction reactions. They are involved in hydride ion transfer. And it's gotten from where? Nicotinic acid, niacin. Also, one of the vitamin Bs. You see it a lot. The vitamin B3, you see it in a lot of, I mean, when you buy, I think vitamin B complexes and, some multivitamins, you see all these things written on them. Pyridoxal phosphate is one of those coenzymes we will see when we are looking at the metabolism of proteins. They are involved in the transfer of amino groups. So we'll see it in a process we refer to as transamination in the metabolism of proteins. And the inactive form, the vitamin from which you get that is pyridoxine. So it's vitamin B6. And we see if you look at the name, this is pyridoxine. And then you see the coenzyme is pyridoxal phosphate. So it means pyridoxine just gets phosphorylated, a phosphate group gets attached onto pyridoxine to make it active. And we'll see a lot of these phosphorylation processes which are involved in the activation of molecules. Tetrahydrofolate, what you usually refer to as folic acid, here is written as folate, folic acid. You also see why folic acid is prescribed a lot, particularly for pregnant women. We'll see the function of it by the time we finish. And it's involved in the transfer of one carbon groups. So a group, it can be some methyl aldehyde group, is just one carbon in there. Because those carbon groups are involved in many reactions into cell proliferations, so development of parts of organisms and things like that. We have thymine pyrophosphates, which is involved in the transfer of the aldehyde group. It's coming from vitamin B1, vitamin B1. So these are, common coenzymes. And you see clearly everything we're talking about here. Virtually everything we've talked about, they are all coming from different vitamin Bs, So that should tell you why, a lot of times when, a patient goes to the hospital, somebody is not well, is prescribed with some medicines, and then is given some B complex in addition. I mean, if you're not well, it means the body is broken down in a way, and medications are being given to you. Those medications require, some metabolic processes. They need to be metabolized in order for that medication to carry out its functions. So the body will need some boosting. So these coenzymes are required by many of these enzymes that are involved in the metabolism of these drugs. Not only the drugs per se, but in that condition of a sickness, you need some boosting of enzymes, system, different other things. So vitamin Bs are, you can see, we are seeing different forms of vitamin Bs that are acting as coenzymes. So you have a better understanding of why it is so. Now, as we do all the time, what we do is we take a coenzyme, we take a look at the structure, and then we look at the function. So we're starting with thymine pyrophosphate. Thymine pyrophosphate. Now, we said thymine pyrophosphate is the active form of vitamin B1, that is thiamine. On the right-hand side, we see the structure of thiamine. So that is vitamin B1. So you see at the end of that vitamin B1 or the thymine structure, we see a hydroxyl group. There's a hydroxyl group at the end. In place of that hydroxyl group, we see that thymine pyrophosphate is having a phosphate group. So as a matter of fact, everything else between thymine pyrophosphate and thymine is the same, except for these two phosphate groups that have been attached onto the molecule, which is to say that the vitamin gets phosphorylated to become active. And what's the meaning? Pyrophosphate. Pyrophosphate, you can say diphosphate. So pyro also means two, di. So two phosphate groups are attached onto thymine for you to have the active form of the vitamin thymine. Now this ring, there is the ring in the middle. You see that same ring also in the structure of thymine is referred to as the thiazolium ring. Thiazo. So you have thiazo. Why? So you have thia. Thia is coming from the sulfur. Azo, the nitrogen. So you have thia, sulfur on the first position, nitrogen on the third position. Thiazo. So you have thiazolium ring. And we see there's a carbon number 2, which is red with a hydrogen on it. Now, the reason we have that red and hydrogen on it is because actually the thiazolium ring is referred to as the active part of this whole structure, the thymine pyrophosphate. As we go ahead, you come to see that it is on this ring that the reactions that are being catalyzed by an enzyme where thymine pyrophosphate is acting as a coenzyme, it is on this thiazolium ring that the reaction is occurring, and particularly on carbon number 2. So this is the reactive part of the ring. The carbon, this hydrogen, is going to react, and on this carbon, you have a molecule sitting and you have a reaction proceeding. Right. Now, we look at the function of thymine pyrophosphate. We saw earlier that it serves as a coenzyme to the enzyme pyruvate decarboxylase. So the name tells you that there is going to be a removal of a carboxylic acid group or a carboxyl group from pyruvate. So pyruvate is a substrate from which the carboxyl group will be removed. And you see this kind of reaction in carbohydrate metabolism, in anaerobic metabolism of carbohydrates, what results in ethanol fermentation, or say carbohydrate fermentation into ethanol. You first of all will have a process of decarboxylation. Carboxyl group is removed, and then you have a further reduction reaction. So what you see here, the bond that is broken by pyruvate decarboxylase is shown in red in pyruvate. So that bond, which is between the carbonyl groups, see the carboxyl group will be broken off. Actually, that carboxyl group is what you see as CO2. What is referred to as a byproduct of a breakdown of, or let's say, metabolic processes that you have carbon dioxide being released as a byproduct. And then you see that a new bond is formed with hydrogen on the residue. Carbon, which is left, there's carbon double bond O. You have a hydrogen now binded onto it. So you now have an aldehyde group. So carboxyl group is broken off, and you now have an aldehyde. In that process, where pyruvate decarboxylase is acting, thymine pyrophosphate is a coenzyme. You have another one, pyruvate dehydrogenase, which acts in the synthesis of acetyl-CoA. So we see again, same pyruvate, the bond between the carbonyl groups broken, and the carboxylic acid group is given out. So that some decarboxylation first occurs. And then after that, so the only difference is that in place of hydrogen, which was bonding on that carbon to give you an aldehyde, you are now having coenzyme A bonding. And so you now have acetyl-CoA. We have alpha-ketoglutarate dehydrogenase. This one too, you see it in the citric acid cycle, the TCA cycle or the Krebs cycle, as you refer to sometimes. And here too, thymine pyrophosphate is acting as a coenzyme. And you see the alpha-ketoglutarate is an intermediate in the TCA cycle. Means similar reaction, the breakdown of this bond between carbonyl groups. Now, we also see transketolase. So this you will see in what we refer to as, carbon assimilation reactions, pentose phosphate pathway, where you have the synthesis of nucleic acids coming from. And clearly what you see here, the bond that is broken is shown in red. And so you see these whole parts, the second carbon with the OH, hydrogen, and R4 is gone. And a new group which is similar to that is bonded onto it. It looks the same, but they are not the same because the R can be different. The side chain can be different. That's why you have R4, but here you have R5. That can be different. So you find this, in the breakdown of bonds that are between carbonyl groups. So you find this like in a decarboxylation of what we refer to as keto acids. We'll see more when we are taking each of these metabolic processes, like in lipids, for instance. We'll come across it somewhere. Now, what we are seeing is the mechanism of action of thymine pyrophosphate. Now, these two, please, it's not a thing that you should cramp in your head. You just have to sort of appreciate the step-by-step process by which reactions occur, so that you don't think that when reactions are written like A plus B is giving you C, it's a simplified way of presenting it, but things that occur in a reaction, if you want to look at it step by step, you see that some intermediates are formed and all of that, and you are supposed to appreciate that, but not to cramp these things into your head. The best you can do for yourself is to learn how to read the reactions and don't be afraid of the structures. So for instance, to read this reaction and understand it, the first thing you should just do, you stay calm and choose an arrow at a time. Here it's made simple. The arrows are numbered. So you can see arrow number 1. That tells you that is one reaction. There's arrow number 2. There's arrow number 3. There's arrow number 4. And then there's arrow number 5. So it's a cyclic reaction that is ongoing. So one arrow at a time, and you'll be able to read the reaction and come to understand what is going on. So the first we are taking, so like we do, you take a reactant and you take the product. Now remember, when we were looking at the structure of thymine pyrophosphate, I said the thiazolium ring is the active part of that molecule. So you see, we are only seeing the thiazolium ring here. The rest of the structure, the phosphate group, will be in this R prime is not being shown. There was another ring that was attached. R inside the R there is not shown. Because if we want to bring all that molecule here, everything will just be clumsy. So we've just brought the active part of the molecule, which is the thiazolium ring, and the rest of the parts of the molecule, we just represent it R prime. Now that understood, you also remember that carbon number 2 with hydrogen on it was red. And I said it was that carbon on which the reaction will be occurring. So here you can see you now have a carbanion because the hydrogen on it is giving out. So that carbon is carrying some negative charge there. And so it can have some nucleophilic attack on the substrate, pyruvate. Pyruvate is a substrate. And what do you see? Pyruvate, now, the product, you see pyruvate is now bonded on the carbon, on that carbon, that active carbon in the thiazolium ring. And the next thing you see is that the carboxylic acid group, this carboxyl group, COO, is removed as CO2. CO2. And you have some resonance intermediates formed. So you see like some double bond between the carbons. Also here you have a single bond formed. So another carbanion. This is a resonance, can be, a reversible reaction. But let's focus and move on to the third reaction from that resonance structure. When we take arrow number 3, we notice that you are having some hydrogen ion coming in there. And that hydrogen ion comes in to bind onto, that carbon, which was having a negative charge on it. So you now see there's a hydrogen that is bonded on that molecule. In the end, you have acetaldehyde. So it's just the reaction which we were looking at earlier in the table, where you take pyruvate and this carboxyl group was removed as carbon dioxide given out. And then there was a hydrogen that came to bind on that carbon to give you acetaldehyde. So now that you have the substrate dissociated from the coenzyme, the coenzyme is free to take another molecule of substrate. So the reaction continues. So the cycle continues on and on. Now, I liken this to, like, if you are in the kitchen, you want to slice some vegetables. You say you want to put the vegetable on a chopping board before you use your knife to chop it. You don't have your vegetable floating in the air and then you'll be using the knife to chase it. So it's like this coenzyme here, thiamine pyrophosphate, is acting like that chopping board on which the substrate pyruvate comes to sit, and then you have your enzyme pyruvate decarboxylase acting on it. All right, now we move on to the next structure of nicotinamide nucleotides. So what you see usually as NAD, NADPH, NADH, or FAD, FADH, FADH2. So those are nicotinamide. No, FAD will be flavin, but nicotinamide will be those NAD, NADH, NADPH. So we're taking a look at the nicotinamide nucleotides. And these two, I mean, we saw already that this was coming from the vitamin niacin, nicotinic acid, one of the vitamin Bs. So if you look at it, this is niacin or nicotinic acid, which is, I think, vitamin B3. And if you look at it, you see that in place. So as a matter of, if this was written not in the ionic form, if it was in a molecular form, this will be OH. There will be a hydroxyl group there. I mean, because a carboxylic acid group there. But I see in place of that, you now have an amine group. That's what makes it nicotinamide. From nicotinic acid, you have nicotinamide. Now, what you see is that this is a dinucleotide. It's a dinucleotide. You see, this is one nucleotide. Below here, you see the ribose sugar, on which you have adenine. And on carbon number 6, you have, did I say carbon number 6? On carbon number 5, you have a phosphate group. Phosphate group. And that phosphate group is bonded onto the phosphate group, which is on the second nucleotide. And that nucleotide, the base, the nitrogenous base on it is nicotinamide. So this is where you have the nicotinamide adenine dinucleotide. So you have nicotinamide as one of the bases, the nitrogenous base. Then you have adenine, so nicotinamide adenine, but the two of them, the two molecules are nucleotides. If you've forgotten what nucleotides are, I think you should go and revise, because this we did also in HND, nucleotides, composed of a 5-carbon sugar, a nitrogenous base, and a phosphate group. Now, in place of this hydroxyl group on carbon number 2, you can have a phosphate group. We'll see. That's what will be giving you what you see as NADP. All right, so that is the nicotinamide adenine dinucleotide structure. Now, these coenzymes are involved in electron transfer from one molecule to the other. In other words, they are involved in oxidation reduction reactions. So we see that oxidation reduction reaction being shown here, where you have some hydrogen ion and coupled with some electrons that are in that reaction, and we see the nicotinamide ring is receiving a hydride ion on it. See the hydride ion, which is in blue, you now see it attached on the nicotinamide ring there. Now here, you pay attention. It's telling you that there is an A side and there is a B side. The molecule, if you are looking at it in a 3D, the molecule has it has a front part, it has a back part. So that we'll come to see some stereospecificity also related to the coenzyme, the nicotinamide nucleotides, where you have some enzymes that are acting with coenzyme that will receive the hydride ion from the front side, and you have some that will receive the hydride ions from the backside. So we see these NAD involved in electron transfer reactions. Call it they are coenzymes to oxidoreductases. So you see them in oxidation reduction reactions. And you can see here, as it receives the hydride ions, it gets reduced. So you now see NADH. That's a reduced form of the coenzyme. Right. So here we're seeing, so what I was talking about, that stereospecificity pertaining to NAD orientation, the orientation of the nicotinamide nucleotide. So we see an enzyme, isocitrate dehydrogenase. If you look at them, all the names, look, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, glucose 6-phosphate dehydrogenase, malate dehydrogenase, glutamate dehydrogenase, glyceraldehyde 3-phosphate dehydrogenase, lactate dehydrogenase, alcohol dehydrogenase. See, all of them are dehydrogenases. And I mentioned in a previous lecture that when you see the name of any enzyme with this dehydrogenase, you should quickly know that is an oxidoreductase. It is an enzyme involved in oxidation reduction reactions. In fact, in some cases, dehydrogenase is a synonym to oxidoreductase. So when classifications are being done of enzymes, oxidoreductases are sometimes referred to as dehydrogenases. And you have, these coenzymes, either the NAD plus or NADPH plus, which will be involved in reduction reactions because they are going to be receiving hydride ions. And you see that some of the enzymes are acting specifically with the coenzyme receiving the hydride ion from the front side, that is the A side, some of them from the back side, which is the B side. So you see that specificity being shown. So that specificity is to the enzyme, which means, for instance, isocitrate dehydrogenase, when it is acting with NAD+, NAD+ will be taking the hydride ions from the front side, not the back side. Whilst you have alpha ketoglutarate dehydrogenase taking it from the backside, as well as glucose 6-phosphate dehydrogenase also taking it from the backside. Malate dehydrogenase will take the hydride ion from the front side, the A side. Glutamate dehydrogenase from the backside. Glyceraldehyde 3-phosphate dehydrogenase take it from the backside. And the last two, lactate dehydrogenase, alcohol dehydrogenase. These two, when they're acting with the coenzyme NAD+, they will take the hydride ions from the front side. So that's another form of specificity pertaining to orientation of the nicotinamide nucleotide coenzyme. This is an example of a reaction. This is pertaining to metabolism of alcohol, but this is going to be some oxidation reaction where you see ethanol. So first ethanol, which is being acted upon by alcohol dehydrogenase to give you acetaldehyde. You have an aldehyde coming from ethanol. And then the reaction continues. That acetaldehyde further undergoes oxidation to give you acetic acid. There too is alcohol dehydrogenase that is still acting. Sometimes when you read this reaction, sometimes the second enzyme is referred to as acetaldehyde dehydrogenase because the substrate is acetaldehyde, whilst the first substrate is alcohol, and so that enzyme is referred to as alcohol dehydrogenase. This is a common reaction with the metabolism of alcohols. You know, one interesting thing about this reaction is that, see, under normal circumstances, when you take homologous series of alcohols, and what I mean is if you take alcohols, depending on the number of carbons that are present in the alcohols, the higher the number of carbons, or as the number of carbons increase in the molecule of alcohol, that alcohol should become more toxic. So for instance, if you say methanol contains just one carbon. Then you come to ethanol. Ethanol has 2 carbons. Then you go to propanol. Propanol has 3 carbons. Go to butanol, pentanol. So that's what I mean by you take a homologous series of alcohols. And as the number of carbons increase, the toxicity of the alcohol should increase. Interestingly, you see, methanol is toxic. It's toxic because, you see this reaction, what it means is that if you take methanol, it will first be oxidized to formaldehyde, which is toxic, be acting on the nervous system, can cause blindness in a short period of time, can cause breakdown of the nervous system. And that formaldehyde, when it gets oxidized further, will give you what? Formic acid, also toxic. Now, from the beginning, I was saying that when the number of carbons increase, toxicity should increase, which means ethanol should be expected to be more toxic than methanol. So how come that humans have alcoholic beverages, which is ethanol based? It's because of the alcohol dehydrogenase acting on ethanol to convert it to acetaldehyde, which is a common reaction in carbohydrate metabolism. And that acetaldehyde quickly gets converted to acetic acid. And once you have acetic acid, this acetic acid can get conjugated to coenzyme A, and you have what? Acetyl-CoA. And acetyl-CoA then goes into metabolic reactions, generating energy. Of course, excessive consumption of the alcohol will result into, say, accumulation of like acetaldehyde. And aldehydes are toxic sometimes. That is what results in some people losing orientation and acting on the nervous system, their heads aching and all of that. Then excessive consumption of it will also result into, bad odor coming from them. So it's an interesting reaction. Right after that, when you go to propanol becomes toxic. Butanol become more toxic, like that. All right. Now we can take the flavin nucleotides. This one too is a dinucleotide. You can see there's a red dashed line, which is separating the two nucleotides. The one below is clear. You have adenine, which is on that 5-carbon sugar with a phosphate group. Then above the red dashed lines, there's another phosphate group. And here you see a straight chain sugar, which is, this time around, it's not in a cyclic structure, but it's a sugar. That's a 5. If you count, the carbons are 5, just like this, the same thing, except that it's represented in a straight chain structure. So the Fisher structure, not the Haworth structure. And then you have the flavin. The flavin. So that's also a nitrogenous base. The 3 rings fused together, what is referred to as the isoalloxarine ring. Now you see clearly that only the top is referred to as flavin mononucleotide, because it's just one, but the 2 of them together is flavin adenine dinucleotide, FAD. So flavin, that is the 3 rings fused together, then adenine. So flavin adenine dinucleotide. This is also involved in oxidation reduction reactions. And we can see clearly here too, hydrogen ions, electrons being transferred. And we see flavin nucleotides being derived from what? Riboflavin, vitamin B2. Okay, now here too we see the enzymes that they serve as coenzymes too. The first 4, they are all dehydrogenases, dehydrogenase. And then we see thioredoxin reductase. This time around, it is specified that it will be involved in reduction reaction. The next one is also a dehydrogenase, NADH dehydrogenase. And we have glycolate oxidase. This one too is specified to be involved in oxidation reaction. And then the flavin nucleotides that will be serving as coenzymes to these enzymes. Either it's a dinucleotide, FAD, or it's a mononucleotide, FMN. And these flavin nucleotides, you find them, bound to proteins that, like, so the enzymes that they will be bonded to as, a coenzyme to, we refer to them as flavoproteins. It's like, that's a prosthetic part, the flavin nucleotide that is binding to the protein, and so we refer to it as a flavoprotein. And you find these flavin nucleotides, acting in processes of what we refer to as the circadian rhythm. You do have some of these in plants as well, as well as in animals. So you see in the morning, you see some plants are going to open up their leaves or their flowers. In the evenings, they close. And mammals, us, in the morning when there is enough light entering to the eyes and all of that, we wake up. In the evenings, We are tired. There is less light. We retire to bed. It's a circadian rhythm. It's a biological clock that works. And these flavoproteins, mediate, the effect of light on living organisms. They also act in, enzymes like photolysis. What did we see as lysis? Involved in either the removal of some groups to form double bonds or addition of groups to remove double bonds. And these type of reactions you find in some photolytic processes of DNA repair in organisms. So you have these flavin nucleotides acting in such processes. Now we take coenzyme A, which is a common one you have been seeing. We'll see coenzyme in many reactions. So we just look at the structure. We don't be talking about the function so much because we'll see it again and again and again in so many reactions. And here too, we see a nucleotide, phosphoadenosine diphosphate. So we see the nitrogenous base, adenine, which is bonded on that ribose 3-phosphate. See a phosphate group on carbon number 3 of the ribose, and two phosphate groups on that nucleotide. And here again, we see one of the vitamin B's, pantothenic acid. We saw pantothenic acid already, pantothenic acid. One of the vitamin B's, vitamin B5 or so, which is shaded in pink. And then attached to it is it's a derivative of cysteine. You remember the structure of the amino acid cysteine? It was having a sulfur hydroxyl group at the end of it. So that's a derivative of for cysteine, the captoethylamine. And here too, we are seeing it is that sulfur hydroxyl group which is the active part of the molecule. So it's shown there that reactive thiol group, the sulfur hydroxyl group. So you see this whole molecule, when it gets attached to acetic acid residue, we call it acetyl-CoA, and that's what you see here. So if you have the acetic group, which is blue, the sulfur hydroxyl group, when it gives out that hydrogen, remember the acetic acid was having a hydroxyl group here, OH. So if that OH binds to that hydrogen, some condensation reaction and the water given out, then this sulfur in the structure of the coenzyme A will now bind onto that carbon to give you acetyl-CoA. So you see why we are always writing it this way, acetyl-CoA. Imagine you have to draw all of this big thing. So you just write acetyl-CoA, make it simpler, but actually this is the complex structure that you are referring to when you say acetyl-CoA. Then biotin, also one of the vitamin B's, vitamin B7. So this is the structure of biotin. It's having some ring structure with some carboxylic acid chain attached onto that fused ring structure. And you see it's involved in the transfer of carboxylic acid group. So we see bicarbonate, the COO group is red. I mean, to track it, to see when it goes on to pyruvate, So this COO group gets transferred onto pyruvate by pyruvate carboxylase. Some energy is used, ATP, and you see the coenzyme biotin. So the structure you are seeing biotin there, you are now here is being written here, biotin, to show that biotin is the coenzyme. And what do you see? That carboxyl group, when it is attached on pyruvate. So here, if you look at the structure of pyruvate, you have a CH3. But you now have CH2, so one hydrogen is gone, and in place of it, you have the carboxyl group attaching onto it, and now you have oxaloacetate. It's actually one of the reactions you see if you just go and take a look at the tricarboxylic acid cycle and see the conversion of pyruvate to oxaloacetate. I think it's even the very first reaction where you see pyruvate acetyl-CoA coming in there. You see formation of oxaloacetate over there. All right. How does biotin do that? So this is the biotin, the two-ring structure fused. What you see here, biotin carrier protein, is only depicting that the enzyme onto which the coenzyme is bonded onto. And you see this is the bicarbonate, what we were just looking at in the previous slide. And we see biotin carboxylase. Here the enzyme here is being referred to as biotin carboxylase, which is transferring the carboxyl group from bicarbonate onto biotin, which is the coenzyme, and biotin is carrying it now onto acetyl-CoA. And the enzyme which is involved in that process of the transfer of the carboxyl group from the coenzyme onto acetyl-CoA is trans-carboxylase. So we see that group, which is shaded green, is now transferred onto acetyl-CoA to give you malonyl-CoA. So, that is biotin involved in the transfer of carboxyl group. What sometimes is just written as transfer of CO2. That's what you are seeing over there. Coenzyme biotin. I think this should be the last coenzyme we're looking at. This is folate, what you usually refer to as folic acid. This too is composed, consisting of three major groups. Glutamate or glutamic acid, that's an amino acid. You have a para-aminobenzoate, that's at the center. And then you have some two fused ring structure, 6-methylpterine. And you see some nitrogens are numbered. They are in blue, nitrogen 5, nitrogen 10, because these are also the active parts of this molecule, the folic acid. That's why they are giving the color, to, if you go looking at, say, maybe mechanism of action and all of that, you realize that it is these nitrogens that are acting in those reactions. Now, I mentioned that folic acid, usually being prescribed to pregnant women because it's involved in lots of proliferation reactions in development. So it's involved in development of particularly the central nervous system of the fetus, what we refer to as the neural tubes, so that the reduction, deficiency of it can result into some nervous system disorders, like the brain will not be developing properly, central nervous system not developing properly, things like that. So a lot of times this is prescribed to pregnant women to prevent such risks. and is involved in the transfer of single carbon groups like methyl group, methylene group, formyl group. That's like an aldehyde group over there, one carbon. So generally, these are referred to as folate-mediated one-carbon metabolism, because all the groups that they are involved in the transfer in contain only one carbon. All right, so that brings us to the end of today's lecture. The floor is open now. Any questions, any contributions, any comments, any concerns? I'm monitoring the chat. You can write in the chat, or you can raise your hand. Then I will enable your microphone. I'll call your name. You unmute yourself. You speak. The floor is open.